I'm Ken Chen, and you can see I am not Swedish. That's the first point. The reason for anyone to come at this time to this booth is perhaps an interest in Stockholm as a site for scientific training, to develop your career, or to consider it as a professional opportunity for your company or other commercial entities. I think this is a region to explore seriously. And I think the way to do this is to compare it to two places I know quite well, because I was in very premier institutions in both places. California, okay, I was at Salk at UC San Diego, worked with Genentech for 12 years as a senior advisor, and in Boston, Harvard, and now here at Stockholm. So the question is why, okay? And what is there here that Boston and San Francisco, who are unequivocally the leaders in stem cell science in general and in biotechnology in the world, why Stockholm? Okay, I mean, that's the obvious question to me. What do we have that might be a little bit better than what they have, because they have everything? So the first point is that for the trainee, your experience is going to need to be very interdisciplinary. It's going to need to be also where you have opportunities to work closely with industry on site in your laboratory, okay? Not you go over to their place and then you don't talk about anything when you come back to your place. There's conflict of interest, there's technology transfer, there are lawyers everywhere, there are barriers, there are silos. Those do not exist for the most part. Doesn't mean there aren't conflict of interest rules, etc. But this is, the rules of the road here are much more integrated to promote hand-in-hand work with industry and with academia. Also, the barriers between clinical medicine and science are much, much less here. If you go to Karolinska itself on the Solna campus, a $3 billion new hospital. Right opposite, a multi-billion-dollar research complex bigger than the Crick Center in London, okay, and a bridge that goes right across them. Next year, in collaboration with Moderna, AstraZeneca, based here in Sweden, and our group, First in Human Modified RNA, will be done here. Designed, made in Sweden.
Just started my own lab, and also, as Ken said, there is quite a bit of funding you can get to do that. And what I work with is that we model neurodevelopmental disorders using reprogramming and iPS cells. Good thing with being here also in Stockholm and Karolinska is this close relation to the Karolinska Hospital. So all my science is of course dependent on me getting hold of patients and patient materials. And also that the patient are well characterized clinically, and also families of patients where it's possible to get samples for reprogramming, both from the patient and from siblings and parents, maybe carrying a mutation or healthy controls. And I think Sweden is, when I talk to colleagues around the world, it doesn't seem as easy for them to get hold of these samples as it is for me, as after I have been setting up good connection with, of course, the clinical scientists, but they are very willing to collaborate, and they do have this register of patients. So I think that's a big advantage, of course, for my research. Even if I do the basic science, I still need to have these collaborations. And then also Karolinska is very strong in the stem cell basic science biology, and especially neural stem cells, which is then my field. And here it's very good to be at the same networks and so on as big professors in the field, where they have described a lot about basic science about neural stem cell and neural stem cell differentiation, etc., which is very important when I want to study the patient neural stem cells and their differentiation. It's very important, of course, to know how the differentiation is in the healthy cases. And this is very good advantage for me when I set up my own lab to actually have connections with big professors in the field that actually have methods maybe set up, or so you can get help with different methods, but also of course for networking and discussions.
The esophageal cancer treatment is we have picked up to move on to the clinical development. And then the Tokyo Women's Medical University team has conducted over 20 clinical research with 20 patients. And they also collaborated with the Karolinska University Hospital team since 2012. And they treated 10 patients. So that means that you take a small piece of the oral mucosa and make a cell sheet. And then after you remove the esophageal cancer with the endoscope, and then you put this cell sheet like a bandage. And that bandage can help wound healing. And by doing so, you can prevent the stricture that can often happen after the surgery. So that quality of life of the patient will be much improved. So after getting a very positive results, the physician team here at the Karolinska University Hospital was very much encouraged us to start the clinical study in Sweden. So we did. So we have established a Swedish subsidiary, Cellseed Sweden AB, in May. And with the help of the people here, and we had a very good, now forming a very good team to get started the clinical study very soon. And we are very much looking forward to it.
So our interest is particularly in mesenchymal stromal cells, and we've been focused on the clinical translation maybe of the cells. So for the last 15 years we've studied mesenchymal stromal cells, and mainly from bone marrow, but also from other tissues. We had a particular interest in their immune modulatory properties and how they affect the immune system, and both in healthy individuals and healthy lymphocytes, but also in diseases. And Ellen, for example, is here and has an interest in how the stroma may contribute to the pathogenesis of multiple sclerosis. So our initial clinical studies were on graft versus host disease, which is an immunologic complication after hematopoietic stem cell transplantation. But over the last couple of years, we broadened our concept, but staying with the immune suppressive and anti-inflammatory mechanism of MSC mediating improvement of disease. So we now continue trials on transplanted patients with severe graft versus host disease, both the acute and the chronic form. And we also study whether MSCs can improve the outcome of newly diagnosed diabetes together with Uppsala University, and improve MS, and also looking at the MSCs in the different disease categories and whether they function abnormally and therefore have changed properties that influence the disease pathogenesis.